Hello everyone, let's learn about the relationship between carbohydrate intake and diabetes mellitus. We are from Group 1 and here is the list name of our team members. Firstly, what is carbohydrates? Generally, carbohydrate is a type of sugars, starches, and fibers that can be found in food. Plants are the primary source of carbohydrate. Meat also may be categorized as part of carbohydrate as it contains sugar that called lactose. Let's move on to the common food sources of carbohydrates. Firstly is grains such as rice, wheat, barley, and also corn. Other than that is legumes such as beans, peas, and lentils. Fruits and vegetables like carrots, watermelon, and broccoli. And lastly, other types including table sugar and honey. Classification of carbohydrate. Carbohydrate can be classified into two main types, which are simple and complex carbohydrate. The two types of simple carbohydrate are monosaccharide and disaccharide. The simplest sugar monosaccharide contain only one sugar unit such as glucose, fructose, and galactose. Combining two saccharide will form a disaccharide such as sucrose, lactose, and maltose. Disaccharide. First, sucrose is made up of one molecule glucose and fructose. Lactose is made up of one molecule glucose and galactose, while maltose is made up of two molecules of glucose. For complex carbohydrate, they are oligosaccharide and polysaccharide. Oligosaccharide consists of 3 to 6 monosaccharide units. The two most common are raffinose and starchose. Next, for polysaccharide, it contains many monosaccharide that link together. For example, starch, glycogen, and fiber. Starch. The simplest form of starch is the linear polymer amylose, which is a single glucose unit, while amylopectin is the branch form that link by alpha-1,4 glycosidic bonds and alpha-1,6 glycosidic bond. So here is the summary of classification of carbohydrates.
Now, let's move to digestion of carbohydrate in our body. The mechanical and chemical digestion of carbohydrate begins in the mouth. The chewing process will tear the food into small pieces. During the chewing action, salivary glands secrete saliva, which contains salivary amylase enzymes. These salivary amylase enzymes break down the amylose and amylopectin into smaller chains of carbohydrate called dextrin and maltose. The partially digested food is called bolus. The bolus travels through the esophagus to the stomach. In the stomach, the acidic environment inactivates the amylase enzymes. Thus, the starch digestion will not occur within the stomach. Then, the chyme will travel to the duodenum. Once the chyme begins to enter the duodenum, the pancreas secretes pancreatic amylase into the duodenum. The pancreatic amylase continues to break down the dextrin into shorter carbohydrate chains. After that, in the ileum, the maltase enzyme hydrolyzes the maltose into glucose. The sucrase enzymes hydrolyze sucrose into fructose and glucose. And lastly, the lactase enzymes hydrolyze lactose into galactose and glucose. Next, absorption of carbohydrate into the bloodstream. The cells in small intestine have membranes that contain many transport proteins in order to get monosaccharides and other nutrients into the blood, then distributed to the rest of the body. Fructose is absorbed by facilitated diffusion through glucose transporter 5, while glucose and galactose are actively transported by sodium glucose transporter 1. On the other side of enterocyte cell, monosaccharides are transported out of the cell through glucose transporter 2 into the blood, where they travel to the liver. The liver is the first organ to receive glucose, fructose, and galactose. The liver takes them up and converts galactose to glucose, breaks fructose into even smaller carbon-containing units, stores glucose as glycogen or exports it back to the blood. Metabolism of carbohydrate includes glycolysis, gluconeogenesis, glycogenolysis, and glycogenesis. Glycolysis is a pathway by which glucose degrades into lactate. Now, let's move to the first metabolism, which is glycolysis. The process starts with glucose and ends with pyruvate. As shown in the video, the flow of glycolysis with enzyme involved from early process until the end. Second, gluconeogenesis. Gluconeogenesis is a reverse action of glycolysis. It starts with pyruvate and ends with glucose. As shown in the video, the flow of the gluconeogenesis with the enzyme involved from early process until the end. Gluconeogenesis is actually defined as a pathway by which glucose is generated from pyruvate or lactate. For glycogenolysis, it is a process by which glycogen is broken down into glucose to provide immediate energy and to maintain blood glucose levels during fasting. While glycogenesis is a pathway by which glycogen is synthesized from glucose. Glycogenolysis starts from glycogen converted by phosphorylase and debranching enzymes into glucose 1-phosphate and to glucose 6-phosphate by phosphoglucoamidase, lastly converted by glucose 6-phosphate into glucose. The reverse action of glycogenesis is known as glycogenolysis. Glucokinase is needed to convert glucose into glucose 6-phosphate. The same enzyme, phosphoglucoamidase, converts glucose 6-phosphate into glucose 1-phosphate, followed by UDPG phosphorylase and glycogen by glycogen synthase and branching enzyme.
Next, we will discuss about the relationship between carbohydrate and diabetes mellitus. As you all know, all carbohydrates are converted into glucose in the body. In a normal mechanism, when blood glucose level increase, the pancreas secretes insulin to convert glucose to glycogen. It will increase the metabolism of glucose in cell. Next, what is diabetes? Diabetes is a disease where sugar levels in the blood is presently high and overflows in the urine. Do you know sugar consumption is not a direct cause of diabetes? There are a variety of causes for diabetes, but none of them reported that sugar is one of them. Obesity is the major causes of diabetes. Sugar consumption is a primary contributor, which is a risk factor for diabetes. As a result, eating too much sugar may lead to diabetes indirectly. There are two main types of diabetes, which are type 1 and type 2. Type 1 diabetes is also known as insulin-dependent diabetes. It is an immune system disorder that has inability to make insulin. Meanwhile, type 2 diabetes develops due to insulin resistance. It happens when some parts of the body such as liver, muscles and fats are not very good at responding to insulin and can't easily take up glucose from blood. As a result, the pancreas produces more insulin to help glucose enter the cell that cause overwork. Then, it can eventually lead the beta cell in pancreas to atrophy and stop producing insulin effectively. There are many types of laboratory tests used to diagnose diabetes mellitus, such as random blood sugar, fasting blood sugar, and also hemoglobin A1C. These tests will measure the glucose level in people's blood. The patient considered to have diabetes when random blood sugar is higher than or equal to 11.1 millimoliter, fasting blood sugar above and equal to 7 millimoliter, and hemoglobin A1C is greater or equal to 6.5%. So that's all from us. Thank you for watching. Here are the references that we used to complete this task.